Hello and a very good day to everyone. I am Joshua Low Juni, a student in the Master of Science in Molecular Medicine program, and today I will be presenting my assignment titled Unravelling Parkinson's Disease from Pathogenesis to Molecular Therapy. Now, let's begin with the introduction. Parkinson's disease is a progressive neurodegenerative disorder projected to affect over 25 million individuals globally by 2050. It is primarily categorized by the degeneration of dopaminergic neurons in the substantia nigra pars compacta of the midbrain and the accumulation of misfolded alpha-synuclein aggregates forming Lewy bodies. PD often extends to other brain regions and involves non-dopaminergic neurons as it progresses. Onset typically occurs at the ages of 55 and 65. With its rising prevalence, there is an urgent need for improved early diagnosis, treatments, and disease management. Parkinson's is diagnosed based on motor symptoms like tremors, bradykinesia, and rigidity, though non-motor symptoms such as depression, anosmia, constipation, and sleep disturbances often appear earlier and reduce quality of life. Table 1 outlines the 5 stages of Parkinson's progression, each representing a gradual worsening of symptoms. Sporadic Parkinson's is associated with environmental toxins, head trauma, aging, and genetic susceptibility. Alternatively, familial PD, typically early onset, is caused by inherited mutations in PARC genes following Mendelian inheritance. Although treatments are mainly palliative, recent advancements have improved the understanding of Parkinson's mechanisms and revealed molecular targets for therapy. Hence, this presentation will reveal the Parkinson's pathophysiology, diagnostics, and emerging molecular therapies. Now, onto the pathophysiology. To understand Parkinson's pathology, we must first examine the nigrostriatal pathway. This pathway involves dopaminergic neurons from the SNPC projecting to the striatum and regulates voluntary movement. Dopamine stimulates the direct pathway via D1 receptors and inhibits the indirect pathway via D2 receptors, maintaining motor control. In Parkinson's, SNPC degeneration reduces dopamine levels, leading to the underactivation of the direct and overactivation of the indirect pathway. This disrupts the thalamocortical signaling and causes motor deficits. Figure 1 illustrates this imbalance where reduced dopamine levels lead to motor deficits observed in Parkinson's, resulting in the underactivation of the direct pathway and the overactivation of the indirect pathway, ultimately causing dysregulation of the nigrostriatal pathway. Parkinson's pathogenesis involves several mechanisms including alpha-synuclein aggregation, impaired protein clearance, mitochondrial dysfunction, oxidative stress, endoplasmic reticulum stress, and neuroinflammation. Alpha-synuclein is a soluble neuronal protein involved in synaptic transmission and mitochondrial function. Under stress or SNCA mutations, it misfolds into the beta-sheet-rich aggregation-prone form. This forms toxic oligomers, protofibrils, and Lewy bodies and can propagate in a prion-like manner, spreading transneurally to other central nervous system regions. The aforementioned process is depicted here in Figure 3. It is hypothesized that the alpha-synuclein aggregates play a central role by initiating a cascade of pathological events. As depicted in Figure 4, they impair alpha-synuclein physiological functions and trigger various downstream mechanisms that promote neurodegeneration, including the induction of synaptic dysfunction, mitochondrial impairment, and neuroinflammation. Next includes the dysfunctional protein clearance systems that lead to the accumulation of misfolded or dysregulated proteins like pathological alpha-synuclein. As seen here in Figure 5, these clearance systems include the UPS system, which involves ubiquitinating abnormal proteins and subsequently directing them for proteasome for degradation, and the LLP pathway, which facilitates clearance via 3 distinct processes, including autophagy, microautophagy, and chaperone-mediated autophagy. Oxidative stress and alpha-synuclein oligomers impair proteasome activators, lysosomal membrane proteins, and molecular chaperones, thereby promoting a vicious cycle of alpha-synuclein accumulation and clearance failure in sporadic cases. Genetic factors are also observed, such as GBA1 mutations, which lead to ALP dysfunction by impairing the GCase1 activity. As seen here in Figure 6, a decrease in GCase1 activity impairs lysosomal membrane integrity and acidification, ultimately leading to ALP dysfunction. Conversely, familial Parkinson's cases involving mutations in various PARC genes, as listed below, have been linked to UPS and ALP dysregulation by increasing cellular stresses, impairing their components dysregulating environmental pH, and etc. Now, let's move on to mitochondrial dysfunction. It is a salient feature in both sporadic and familial Parkinson's cases, contributing to dopaminergic neurodegeneration and chronic ROS degeneration. In sporadic cases, environmental exposure to neurotoxins or pesticides may induce mitochondrial dysfunction by inhibiting the mitochondrial complex 1, which leads to impaired ATP production and elevated reactive oxygen species generation. ATP deficiency compromises essential neuronal functions, whereas excessive reactive oxygen species levels induce oxidative stress to mitochondrial DNA, lipids, and proteins, further exacerbating the mitochondrial impairment. This pathological synergistic effect promotes mitochondrial permeability transition pore opening, triggering both dopaminergic neuronal apoptosis and neuronal inflammation as depicted here in figure 8. Similarly, alpha-synuclein oligomers bidirectionally exacerbates mitochondrial impairment by interacting with mitochondrial membrane and inhibiting the complex 1. This disrupts mitochondrial calcium homeostasis, which induces mitochondrial swelling, depolarization, and cytochrome C releasing, leading to apoptosis. Additionally, as neurodegeneration occurs, dopamine leakage and oxidative metabolism generate toxic metabolites, further exacerbating the neurodegeneration and mitochondrial impairment. In familial cases, however, mutations in certain PARK genes, including the PINK1 Parkin, DJ1 and LRRK2, lead to the impaired mitophagy, reduced mitochondrial antioxidant activity, and excessive mitochondrial fission, thereby promoting oxidative stress, neuroinflammation, and neurodegeneration. Furthermore, endoplasmic reticulum stress has also been linked to Parkinson's progression. Pathological alpha-synuclein accumulation, impaired calcium transfer balance between the ER and the mitochondria, and elevated ROS levels collectively trigger ER stress. This initiates the UPR, restoring ER hemostasis by attenuating global protein synthesis, upregulating the molecular chaperones that assist in protein folding, and increasing the degradation pathways. Nevertheless, as illustrated here in figure 9, chronic UPR activation shifts the response from adaptive to pro-apoptotic, ultimately exacerbating the neurodegeneration and accelerating PD's progression. Collectively, these pathological mechanisms drive neuroinflammation. Misfolded alpha-synuclein, acting as damage-associated molecular patterns, activates microglia initially to clear debris. However, chronic activation shifts it to become a pro-inflammatory M1 phenotype, which releases reactive oxygen species, nitric oxide, and cytokines. This also activates astrocytes, perpetuating the neurodegeneration. In turn, neuronal debris released during neurodegeneration perpetuates glia activation, establishing a vicious inflammatory cycle. Genetic mutations contribute to neuroinflammation as well, where SNCA, PINK1, Parkin, and GBA1 mutations promote alpha-synuclein pathology. Alternatively, DJ1 mutations impair the antioxidant defenses, leading to oxidative stress-induced inflammation, whereas LRRK2 mutations promotes pro-inflammatory microglial activation. Figure 10 shows the vicious cycle of neuroinflammation observed in Parkinson's initiation and progression, as aforementioned. Notably, neuroinflammation promotes lipid peroxidation by disrupting the iron homeostasis and elevating the ROS levels, ultimately triggering ferroptosis, an iron-dependent form of cell death that exacerbates the neurodegeneration. Recently, gut microbiota has also garnered significant interest for its critical role in neurological disorders, primarily due to the gut-brain axis. Microbiome dysbiosis have been linked in several Parkinson's cases where it's theorized to initiate Parkinson's. Overgrowth of certain microbial taxa demonstrated enhanced pro-inflammatory cytokine secretion and promote alpha-synuclein aggregation. This may translocate to the central nervous system, ultimately driving pathological changes in the brain. Subsequently, let's discuss on the Parkinson's diagnostics, as timely and effective therapeutic interventions depend heavily on the early and accurate diagnosis. Clinical examination remains the gold standard for Parkinson's diagnosis based on core motor symptoms supported by non-motor features and the exclusion of atypical signs. Despite up to 80% accuracy, tools like the MDS UK criteria often lead to late diagnosis, offer limited pathophysiological insights, and overlap with other Parkinsonian syndromes. Alternatively, neuroimaging plays a supportive role where tools like CT scans and MRI assess structural alterations of the brain, helping to exclude other causes and aiding its differentiation with atypical and secondary Parkinsonism. However, their sensitivity for idiopathic Parkinson's is relatively low. Therefore, more advanced functional imaging modalities, particularly the DART SPECT, have been developed. It utilizes specific radionuclide traces to visualize the dopaminergic function, primarily through assessing the presynaptic dopamine transporter density, enabling greater sensitivity and specificity in identifying the nigrostriatal degeneration. Nevertheless, it remains unreliable in distinguishing Parkinson's from other neurodegenerative Parkinsonism. Fluid-based biomarker discovery represents a key advancement in Parkinson's diagnosis, potentially supporting early diagnosis, monitoring disease progression, and providing deeper insights into Parkinson's pathophysiology. Cerebrospinal fluid remains the best source due to its direct contact with the brain's extracellular space. One of the most promising fluid-based biomarkers includes the oligomeric to total alpha-synuclein ratio, showing correlations with longitudinal disease progression and motor symptoms deterioration. An emerging tool that may aid in this detection includes the RT-Quick ultrasensitive protein amplification assay, which facilitates early and more precise differentiation from PD from other synucleinopathies with a sensitivity and specificity of 95.3% and 98% respectively. Blood-based biomarkers are non-invasive alternatives under investigation, though systemic variability and low concentrations remain a challenge. Figure 13 depicts notable fluid-based biomarkers for Parkinson's in addition to pathological alpha-synuclein, including the elevated inflammatory mediators, increased RRK2 kinase activity, and reduced GCase activity. Genetic phenotyping is also becoming an increasingly feasible and valuable diagnostic tool for Parkinson's. This technique involves screening for Parkinson's-associated gene mutations, primarily the PARC genes, in high-risk individuals, thereby enabling early diagnosis, risk assessment, and patient stratification. Lastly, AI-driven technologies have shown promise by enhancing its accuracy, speed, and objectivity of Parkinson's diagnosis and management. This includes smartwatches with built-in initial sensors that continuously record subtle motion data like tremors and bradykinesia and non-motor patterns including sleeping patterns, two computerized analysis of voice and speech patterns which can detect subtle changes in vocal features such as dysphonia and hypophonia with an accuracy of up to 99%. Additionally, by implementing machine learning and deep learning approaches, AI can rapidly and accurately integrate large-scale diagnostic data, ultimately facilitating in differential analysis with other Parkinsonal syndromes, uncovering hidden patterns, generating predictive models that enable patient stratification. Below demonstrates one of the AI-driven technologies for Parkinson's management, where the motion data streams detected by the smartwatch are analyzed via machine learning algorithms and subsequently transmitted real-time reports directly to the smartphone or healthcare professionals. Penultimately, let's discuss Parkinson's molecular targets and recent therapeutic innovations. Currently, there is no known cure for Parkinson's, as all established therapeutics remain palliative in nature. These include dopaminergic therapy, monoamine oxidase inhibitors, and anticholinergics, which aim to reduce motor deficits by restoring the dopamine signaling, whereas therapies like antidepressants aim to reduce the non-motor deficits like depression. Additionally, surgery are also well established, but may lead to irreversible adverse effects. Nevertheless, they do not halt disease progression or address the underlying neurodegeneration, prompting the pursuit of novel disease-modifying therapeutics that target the specific molecular pathways. A notable emerging therapeutic includes the anti-alpha-synuclein, which aims to restore the alpha-synuclein homeostasis. One approach includes reducing its production, as seen in UCB0599, which directly interacts with the aggregation-prone regions of the monomeric alpha-synuclein. This stabilizes its native conformation, preventing misfolding and subsequent toxic aggregation. Another approach includes enhancing its clearance or inhibiting its prion-like cell-to-cell transmission through immunotherapy. Passive immunization employ monoclonal antibodies such as prasinezumab to target aggregated alpha-synuclein and aid its immune-mediated clearance via microglial activation, whereas active immunization implements synthetic peptide vaccines like UB312 to stimulate adaptive immune response against pathological alpha-synuclein. Overall, both strategies were well tolerated and reduced pathological alpha-synuclein levels. However, they demonstrated only modest clinical benefits, which and were associated with adverse effects, warranting further validation. The last strategy involves modulating the underlying pathological pathways that contribute to the misfolded alpha-synuclein accumulation. A notable example includes the selective LRRK2 kinase inhibitors like DNL201, which counteracts LRRK2 hyperactivation. It showed favorable safety profile and lysosomal stabilization in Parkinson's patients in clinical trials. On the other hand, Ambrosol, an antitussive agent, demonstrated to enhance GCase levels by acting as a pharmacological chaperone, potentially reversing ALP dysfunction and reducing alpha-synuclein accumulation. As neuroinflammation is directly proportional to dopaminergic neurodegeneration, anti-inflammatories were also proposed. This includes the NLRP3 inhibitors, particularly the MCC940, which counteract the neuroinflammatory cascade triggered by the NLRP3 inflammasome assembly post-microglial activation. MCC940 showed promising preclinical results where it reversed inflammasome inactivation, thereby alleviating motor deficits, reducing dopaminergic neurodegeneration, and decreasing alpha-synuclein accumulation. Furthermore, as the JAK-STAT cytokine signaling pathway plays a central role in neuroinflammation, its pharmacological inhibition was also explored. Notably, AZD1480, a selective JAK1/2 inhibitor, demonstrated a reduction in both microglial activation and pro-inflammatory cytokine release in rodent models overexpressing alpha-synuclein, ultimately mitigating the neuroinflammation. Below demonstrates the downstream effects upon NLRP3 inflammasome activation that triggers neuroinflammation and subsequently Parkinson's. Cell-based therapy represents another promising Parkinson's therapeutic approach, aiming to restore dopaminergic function by replacing lost or dysfunctional neurons. A well-documented approach, due to it being safe and ethical, involves the implementation of induced pluripotent stem cells, which are capable into differentiating into dopaminergic neurons. Preclinical studies in primate models have demonstrated that iPSC-derived dopaminergic neurons can survive long-term post-transplantation with minimal immunological response, further supporting their therapeutic potential. Gene therapy has also emerged as a promising therapeutic approach for Parkinson's, aiming to replace, correct, or silence pathological genes. A strategy involves delivering a functional GBA1 gene to restore GCase expression in PD patients harboring GBA1 mutations. This showed promise as an AAV9 vector-based gene therapy encoding the wild-type GBA1 gene, alleviated neuroinflammation, and reduced alpha-synuclein aggregation by increasing GCase activity in the PD-induced animal models. Additionally, gene delivery of neurotrophic factors, which are peptides that support neuronal growth, maturation, survival, and regeneration, led to the enhanced neuronal function and increased fiber density within the striatum in PD-induced animal models. Moreover, the CRISPR-Cas9 technology have also been exploited in preclinical studies to enable gene editing of PD-associated mutations. Notably, silencing the mutant SCNA gene significantly reduced alpha-synuclein overexpression, microglial activation, and dopaminergic neurodegeneration in rat models. Similarly, correcting LRRK2 mutations in patients-derived iPSCs successfully restored physiological gene expression and cellular function. Nonetheless, human clinical trials are essential to evaluate their efficacy and safety. This graphical illustration depicts Parkinson's disease-associated genes that can be edited using the CRISPR-Cas9 technology with the aim of developing Parkinson's models and identifying potential therapeutic targets or strategies. With the recent association of gut dysbiosis with Parkinson's pathogenesis, fecal microbiota transplantation has garnered pharmacological interest in potentially treating Parkinson's. It involves purifying stools from healthy donors and introducing them to patients' GI tract, aiming to re-establish a diverse, balanced gut microbiota. Preclinical studies have demonstrated that FMT can attenuate neuroinflammation in PD-induced animal models, whereas clinical trials have demonstrated improvements in motor and non-motor symptoms in PD patients. These therapeutic effects are largely attributed to the enhanced microbial diversity and restoration of gut homeostasis, positioning FMT as a promising therapeutic candidate for Parkinson's. Finally, let's summarize and conclude this presentation. In conclusion, Parkinson's is an ever-growing progressive neurodegenerative disorder driven by the interplay of environmental and genetic factors. These converge into several shared pathogenic pathways which collectively drive dopaminergic neurodegeneration and a wide spectrum of motor and non-motor deficits. While the current diagnostics predominantly focus on identifying core motor manifestations, advances in molecular imaging, multi-omics-based biomarker discovery, and AI offer a promising avenues for earlier detection and improved patient stratification. These may facilitate the identification of at-risk individuals prior to the irreversible neurodegeneration, supporting timely therapeutic interventions. Moreover, stratifying patients according to their specific underlying molecular pathologies supports precision medicine implementation, ultimately allowing more tailored and efficacious treatment approaches. In parallel, emerging disease-modifying therapies aim to target the underlying pathological pathways of Parkinson's rather than solely alleviating the symptoms. AI integration into therapeutic development and clinical decision-making may also enhance the predictive modeling, therapeutic optimization, and real-time disease monitoring. Nevertheless, continued interdisciplinary research remains vital to elucidate PD pathophysiology, validate the robust biomarkers, and translate preclinical breakthroughs into effective and safe clinical applications. Ultimately, this would enable a paradigm shift towards precision medicine, potentially improving outcomes and the quality of life of Parkinson's disease patients. And with that, I would like to thank everyone for their attention, and I'm happy to take any questions. Here is the list of references.